Hey everyone, this is Mrs. Mallard here, and this is a short lesson to cover the topic of cloning. So we're going to talk about what a clone is, and also look at two different types of cloning: reproductive cloning and therapeutic cloning. So first of all, let's start off with what a clone is. And simply, a clone is just an organism that has the same DNA or is genetically identical to another organism. So I took an example here. We've got an amoeba, and the amoeba has simply divided into two. So this cell would have the same DNA as this cell over here. So we would say that our amoebas here are clones of each other. All right, another example of a clone is if you had a plant and you cut off a section of that plant in order to grow another plant. Both plant one and plant two would have the exact same DNA, so we would say that they are clones, that because of the fact that they are genetically identical. Same thing with bacteria. Bacteria divides into two. Bacteria 1 and bacteria 2 has the same DNA. They are genetically identical, so we would refer to them, we would say that they are clones. All right, so that is our biological definition of what a clone is. Now we're going to go ahead and move on and talk about two categories of cloning. One type of cloning is referred to as reproductive cloning. And in this type of cloning, our objective is we want to make uh, a completely new organism, if you will. So it's kind of like we're trying to make a copy of an organism. So, for example, there was a TV series, I think it was on TLC, about people wanting to clone their pets. So, an example of reproductive cloning would be your cat dies and you want another copy, so to speak, of your cat. So you would do what's called reproductive cloning in order to create another cat, and then of course the new cat would resemble your old one. Okay, that's what reproductive cloning is. The other type of cloning, therapeutic cloning, is we don't want to make a whole separate organism. We don't want to make a whole separate organism. However, we want to clone to create cells and then use those cells as a medical treatment. Okay, so for example, an example of therapeutic cloning would be that we would want to clone to create stem cells to maybe fix someone who has a damaged spinal cord. So in this scenario, maybe someone got in a car accident, maybe their spinal cord was severed or there was damage. Our goal for therapeutic cloning is we will want to clone the person's cells in order to create stem cells to use as a possible medical treatment. So that's the difference between reproductive and therapeutic cloning. Now, for reproductive cloning, the famous famous example here, and this is the first mammal that was cloned, is Dolly the sheep. So you might have heard about Dolly the sheep. This was a big deal back in the late 90s because Dolly was the first mammal. So scientists had cloned things like frogs, for example, but Dolly was the first mammal. All right, and that's why this was a big deal. So let's take a look at this example of reproductive cloning. So in this example, we take an egg cell from a female sheep. So there's my egg cell there, and I'm taking a somatic cell, a body cell from another sheep. And in this case, they took the cell from the udders of the sheep. All right, so this is a body cell. In the egg cell, we take the nucleus out. Remember, egg cells only have half the amount of genetic material, so they're haploid cells. If this were a human cell, this cell would only have 23 chromosomes. Our body cell, on the other hand, the nucleus has a full set, has 2 sets of chromosomes. It's referred to as a diploid cell. So if this were a human cell, we would have 46 chromosomes in here, 23 in here. All right, so our first step kind of is we have our egg cell and our donor cell. We're going to take the nucleus out of this egg. So we're going to basically discard the nucleus. Our next step is we want the nucleus from this body cell that has two sets of chromosomes. We want to get that nucleus into basically this empty egg, and we do that. We kind of shock it so that the nucleus goes in. Now I have an egg cell with a nucleus that has the two full sets of chromosomes, all right, from this sheep right here. And we're going to make sure that this new cell is going to start dividing. So that cell is going to start dividing to produce an embryo. Now, the embryo, we just can't let it keep growing in a petri dish. That embryo has to go into a female sheep where it can continue to grow and develop into a lamb. And of course, this sheep is going to give birth eventually to that lamb. And so now we have our new organism. This is our lamb that was born, and this lamb right here is a clone of the sheep that we got the nucleus from. All right, so this sheep right here and this one right here, if we were to check the DNA from this sheep and this sheep would be exactly the same. So most of kind of where we're at now, we don't see a whole lot of examples of reproductive cloning because they found that organisms reproduce through this method. Sometimes they have a shortened lifespan. Maybe they are born with birth defects. Sometimes the success rate is low. So it took like over 400 tries for them to get Dolly. So these are kind of all negative things. The only I guess, positive thing for reproductive cloning is that the organism is going to look like the organism that it was cloned from. Therapeutic cloning, looking at kind of some of the pros and cons, is we talked about cloning to get stem cells to treat medical issues. The other kind of up and new kind of thing, up and coming thing, is this idea of organ engineering or tissue engineering, where we're using stem cells in order to actually grow like like a bladder in a lab, or growing heart tissue, for example. And once again, these are all things to help maybe somebody who maybe has bladder cancer. Well, you grow a bladder in a lab using their cells, and so then they're able to kind of replace that organ. So that's something that's kind of up and coming. It used to be, when cloning first came out, that embryos had to be destroyed in the process, and of course that was an ethical issue, because the question is do you define an embryo as a living thing or not? And so kind of there was debate around that. Since then, we have figured out other ways to get stem cells. Like, we have learned that there are reservoirs in our body that have stem cells, for example. And so destroying an embryo is not necessary. There also are just kind of ethical, religious kind of issues surrounding cloning. Are we playing God? Are we messing with Mother Nature? Kind of some of those broader philosophical questions. And then the other thing with therapeutic cloning is are there any sort of negative impacts? So, for example, there was a study done with rats where they had spinal cord injuries, and they used stem cells that were cloned in order to help repair the spinal cords of these rats. So that was a positive thing. However, one of the side effects was that it produced multiple tumors in these rats, and so that was a negative impact. And so and that was something that scientists couldn't control. So these are just some of the pros and cons to therapeutic and reproductive cloning. And then the assignment that you're going to work on today, is a virtual lab. In the virtual lab, you are going to simulate cloning a mouse. So in the virtual lab, you are going to simulate the process of reproductive cloning and the steps in order to clone a mouse.